This is a very special event, the Berger Award presentation. This award was established in 1983 to honor Professor Martin J. Berger of MIT and the University of Connecticut, to recognize contributions of exceptional distinction in areas of interest to the ACA. Professor Berger was a mineralogist and early pioneer of many aspects of crystallography. This year's awardee of the M.J. Berger Award is Professor Chiu, the Wallenberg Bienenstock Professor at Stanford University and SLAC National Accelerator Laboratory. It is impossible to overstate the recent revolution in electron microscopy and structural science, most notably single particle reconstruction by cryo-EM. Professor Chiu has made pioneering contributions to the development of biological electron microscopy from its earliest stages. Professor Chiu is the driving force behind the NIH-funded National Center for Macromolecular Imaging at Baylor College of Medicine, and is well known for leading many workshops and as a generous collaborator, including many lowly X-ray crystallographers into this field. Professor Chiu has many well-deserved recognitions, including membership of the National Academy of Sciences. And as the ACA has featured for some time the development of electron microscopic and diffraction techniques in structural science, it is with great pleasure that I congratulate Dr. Chiu on this recognition and invite him to deliver his award lecture. So Professor Chiu, go ahead and get started as soon as you're ready.
David, I thank you very much to bestow me this very prestigious award. I am really honored and thankful to the nominators and the committee to select me this year. And so in the lecture that I'm going to give in the next hours, first is to review some of the historical development in cryo-EM microscopy. And then I will give a number of examples that using the single particle cryo-EM done in my laboratory in the last couple of years. And lastly, I would also present some of the examples how we can extend that from single particles to the whole cell. And so, I'd like to start with my thanks to Professor Bob Glaser, who was my PhD thesis advisor in the mid-70s. And I was, at that stage, I was inspired by the pioneering work done by my colleague Ken Taylor, now for the State University, where he was able to record a electron diffraction pattern of a thin catalase crystal with diffraction spot extended to 3 angstroms. And this suggests that that method by embedding protein crystal in a matrix of ice would be able to preserve the specimen to near atomic resolutions. Following that work, I was inspired to look for protein crystals that diffract in a electron microscope, working with one of my fellow graduate students at Berkeley. We were able to identify a cotoxin complex that formed thin crystals. And so when I put this crystal embedded in glucose, which was an alternative method in preserving the biological specimens in the microscope suggested by Nigel Unwin and Richard Henderson in the 70s, I was able to get electron diffraction pattern with spot extended to 1.6 angstroms. Now, one of the real value of electron microscopy is the ability of forming images. And from the images, one can retrieve the phase information of the objects. And so the diagram on the right is an optical diffraction pattern from an image that I collected together with Jane with the help of Frank Amin at the Fritz Haber Institute in Berlin. At that time, I used a really very state-of-the-art and sophisticated instrument, which was called superconducting lens electron microscopes. So it was a very difficult instrument to operate. Everything was manual. So we managed to collect images and put in an optical diffractometer, saw the spot at 3.9 angstrom. I think this data signify the possibility of recording images of biological specimen at near atomic resolutions. And using this setup in subsequent years, Richard Henderson worked with Samlin to extend the the structure analysis of bacteriodopsin, where it was the first atomic model from two-dimensional crystal by electron crystallography. And those specimens were embedded in liquid helium temperatures. And following that work, Fujiyoshi in Kyoto developed a electron microscope with JEOL company where one could keep the specimen also at liquid helium temperature. And Werner Gruber in Germany and Tom Watts here in Switzerland went to Fujiyoshi lab. They were able to get another 2 sets of near atomic resolution structures of membrane protein. And following that, Eva Nogales and Ken Down at Berkeley used a liquid nitrogen temperature to solve the first tubulin atomic structure. And so all these were had the root of crystallography. That's why many of our colleagues still have an affiliation with crystallography in general. And so since that work, I was inspired by the work of Jacques Dubouchet, who improved the method at Ken Taylor and Bob Glazers in freezing the specimen in a more efficient cryogen, namely liquid ethane, which is the coolant that we use. In such a coolant, we were able to, he was able to cool the specimen to form an amorphous ice called vitreous ice. So I was inspired by that method and began to switch from crystallography to single particle, and very thankful for numbers of really talented postdocs who were the first authors in some of these papers that we started to work with icosahedral viruses. Initially, we were at a kind of 40 angstrom resolution in the 80s. And subsequently, I spent a year at the MRC with Tony Kaufer. We were able to demonstrate the first time on a single particle with Tomiko mosaic virus that we were able to resolve the helices very clearly. And then subsequently in my lab, that with Hong Zhao at UCLA, Steve Luckett at Baylor, and we extended the single particle method to sub-nanometer resolution. I want to point out this is really a lengthy process because at that time the microscope was all manual and the data yield was very slow, and also none of the programs were available at that time. So as we proceed from every few angstrom, we have to develop a new software in that era. So it was a very exciting period that I was able to do that with the support from NIH with my P41 centers. And so, and then later on in 2008, there are 4 structures were published during that year that near atomic resolution single particle were possible. 2 of them from our lab. One we used the cryo-EM to demonstrate that could be done because the crystal structure was known by Paul Signer's group by crystallography. And then we also applied to an icosahedral bacteriophage called Epsilon 15 with Jonathan King at MIT. And so this is really exciting because there's no homolog structures of this virus, of this protein were known at that time. So in this structure, one interesting thing was we were able to build the atomic model of one polypeptide, and there were additional density which could not be modeled. So we actually subsequently did another more biochemical analysis, discovered another protein did exist in this is icosahedral virus. So that's, I think, is one classic case using structural analysis to determine the presence of another protein in macromolecular complex. At the same time, the other group, Nico Gregoria and Steve Harrison work on the rotavirus, and Hong Zhao used to be in our lab, pick up the project on the polyhedral virus. They were able to get the resolution at a kind of 4 angstrom resolution. So I think in the early 2000s, that was really a signal of that single particle is a viable approach to trace a polypeptide backbone from the density map. And since then, there were a big rise in terms of protein of cryo-EM structure deposit in either the EMDB and PDB. Helen Berman, then the director of PDB, worked with me and promote the importance of deposit data in the public domain. And we work together with our colleagues in Europe, in EBI, and also at the Rutgers. So now it become a standard to deposit structure done and in this public archive. So anyone with the internet will be able to access to it. So this is a striking increase since 2008. Our first near atomic resolution structure were demonstrated, and there was a exponential growth, particularly in around the mid 2014 or 15, because of the availability of a new detector, make the whole technology more simpler and more routine, and be able to reserve the resolution of the data that one can retrieve computationally. So among all these structures, many of them actually have a resolution in this orange range between 3 or 4 angstroms, because it turned out to be a really nice resolution that allow one to be able to trace the entire backbone of a polypeptide. And so as you can see in the last few years, the increasing rise of structures at even 2 or 3 angstroms, and some of them actually at resolution better than 2 angstroms. So about 3 years ago, I set up my first Cryo Titan microscope with a K2 camera with a energy filter. It was a very kind support from a shared instrumentation grant from NIH with a matching fund from Stanford. So with this one instrument, we were able in a year and a half to solve 15 structures. Some are protein machine, some are virus and viral protein with antibody complexes. Some is membrane protein and RNA protein complex and RNA. So in the next few minutes, I'm going to select 3 examples to demonstrate what information we can retrieve. And I'll talk about an example of RNA, an example of coronavirus, and then an example of a chaperone complexes. And so now, when we look at the statistics about the archived structures in PDB, it's apparent with the help from Kathy Lawson's, we were able to plot out this curve about the so far the RNA containing macromolecule in PDB. As you can see, that is this plotted the number of structures per year, and in so far, most of the structure were obtained by X-ray crystallography. And until 2 years ago, the cryo-EM became the dominant technique for RNA-contained macromolecules. So I'm going to show the first example about how this is done today, because that was not common a few years ago. And so the example that I like to share with you is the texture harmonized ribosome. And this is the RNA that discovered by Tom Jack, who discovered the RNA can work like an enzyme. It involves a cleaving of the intron and then ligate it and then cleave it again. And so this is an amazing molecule. But there are over several hundreds papers have been published in the literature about the biochemistry, biophysics, and genetic of this enzyme, but none of them report the entire full structures so far. And so we decided with Riju Das in the biochemistry department at Stanford, we decided to just try that out to see what we can see. So these are the typical images of this ribosome, 128 kilodalton, and these on the panel B are the class averages of those particles of the same orientation that we saw that different angular view in projection. With that, we were able to solve a structure with less than 500,000 particles to have a resolution 3.1 angstroms, and this is the density map of such structures. And so here now, here is the exciting aspect that now we can actually put the whole tertiary fold of this intact ribosome, because it turned out RNA is thought to be very flexible and really difficult to crystallize in general. Is also often slightly bigger than what the NMR can handle. So I think is a little niche that the cryo-EM can meet the need. And so this is our tertiary structural fold. And the diagram is the typical secondary structural displays of all the hydrogen bond involved with all these 400 nucleotides. This is the 5' end, this is the 3' end. So and then all are these different types, a dozen different types of hydrogen interaction, which are to the heart of RNA biochemists. And so, in fact, I was so excited that after we did the structure, actually be able to trace the backbone from one end of the molecule to the other end. So from the hand drawing thing from the right hand side to the left hand side, and then up and down, and eventually down again and up, and go all the way. And so finally we get to this peripheral part of the RNA that encompass the catalytic regions. So the next slide, what I'm going to show you the detailed features, what you can see in the density map. So in the middle is the density map superimposed with the model. The one that's shaded with the density are the one that has previous known structural information whatsoever. The middle part in here, which has the model, has different types of crystal structure were solved with small fragments, either with mutated RNA piece or abbreviated one. So the exciting aspects of this is we see these two almost orthogonal helix, two strands that perpendicular to each other that form the perimeter regions of these intact ribosome molecules. And so all these inserts are different types of hydrogen bond interaction. Like, for example, we see the base stacking in these regions, the P2 regions, and then we see these triplex doing the hydrogen bond, and other triplex from right here in these regions, and these quartets and how you see all these hydrogen bond interactions. And so these are really unusual types of interaction that we were able to see to how to hold the catalytic core regions together with the perimeter regions of this RNA. And so here is a movie I want to share with you that we can kind of look side by side from the secondary structure element together with the density map and the model together. So in here, we put on all the different regions as in the secondary structural diagram. And I'm now zooming in here in these two regions here in dark purple and light purple. You can see the base stacking and also the hydrogen bond interactions. And I put the density so you can see the model actually fit the density quite well in practically all the region we interpret. So these are the triple basis interactions. And then in the other side is this really interesting interactions around the region with all kinds of different types of hydrogen interactions, which are very specific chemistry about the RNA molecules. In addition to the Watson and Crick, there's a non-Watson and Crick types of interactions. And then in addition, we were able to pick up large numbers of metallic signals. These magnesium are known to be involved in the catalytic deposit. So we were able to pick up about 30 metal ions in these structures in here. And so in addition to the structure, we in fact we're able to solve another structure with the substrate, which unfortunately I would not have time to talk about today, and it should be coming out in the paper in a week or so in Nature. And so with this in mind, then we also look at the structures, examine the different types of more flexible region of the structures. Here is a way that in cryo-EM using the method developed by Saul at the MRC Cambridge, that one is able to pick up certain regions and do extensive focus classification in such a way that we can see different part of this molecule can have multiple conformations, and they can actually have both a rotation and translation flexibility, particularly in these perimeter regions of this piece of RNA. So that concludes my first example, that one can now use the cryo-EM to solve a atomic model of an intact RNA-only molecules. Now, the second example I like to talk about is a protein folding machine, which we have worked for some time. Back in almost a dozen years ago, we worked with Judy Freeman and Mike Levitt on this chaperone from ACA. And it's a 1 million Daltons, so much bigger than the RNA piece, and has made up of 18 identical subunits, and from 2 rings is different from the more familiar chaperone called GrowEL. GrowEL has a GrowES, but when it does the protein folding inside the chamber, but in this ACA MMCPN, there's no chaperone at all. It's just one single parapeptide. And it's well known that all the folding activity is driven by the hydrolysis of ATP. So from a chemist's point of view, it would be of interest to look at the structures without ATP or with low concentrate ATP that would signify just only the binding before the hydrolysis and also that after the hydrolysis. And so these structures was done at 4.3 angstroms with really an old instrument. And so with our new instruments, as I saw a while ago with the Titan cryos, we're able to extend the resolution by 2 angstroms. You can see that with about 450,000 particles, we can in this close conformation with a larger ATP and aluminum fluoride to engage the chaperone in enzymatic activities. And in fact, we can actually trace the ATP density quite well in here. It's all the side chain and backbones are clearly resolved. So in order to examine how this structure may look differently at a lower concentrate ATP, so we turn in the next biochemical experiment by adding much lesser amounts of ATP with the MMCPN. So in that structures, we find that we can't get on the 2.3 angstrom, we can get around 4 angstrom resolutions. And in addition, a larger density at the apical regions of this map are actually missing. So when we estimate the resolution distribution in this map, all the apical regions around 10 angstrom, whereas in the equatorial domain regions, they can as good as 3.5 angstroms. And you can, some of them you can actually trace out the ATP density as well. And so, so the question that I raised at this point is, okay, they wash out when we average 160,000 particles in this case, but what does it look like in each of these subunits? So the next few slides is to demonstrate how we can get after the conformations of each of these subunits in each MMCPN particles. So the way we did it was to break the 2-ring structure into a single ring and then classify the single ring in multiple classes. So these are the side view, these are the top view, and you can see they are not identical. They look very similar, but they're not identical. Okay, so almost 50% of that have this conformation. So we use this 50% and classify again. The way we classify this time is to computationally isolate each of the single subunit out of this X identical subunit in each of the rings. So that means we have in this we ask for 10 classes. 2 of the classes doesn't look like anything interesting, and 8 classes does look like a single subunit, and it's good enough that we can build a model. And in fact, we examine the enzymatic sites of this subunit. 4 of these structures actually have the nucleotide density, another 4 doesn't. So now, in this, since we started with this 50% of particle and we classify the subunit, so as a result, we know each of the classes which the subunit come from which ring. So because of that statistics, we are able to figure out the number of ATP in each ring. So you can see then that's the proportion of the ring. So the majority of the ring has an ATP 4 to 6 ATP. So that means some ring has only 1 ATP, some are 2, some are 3, some are 4, and up to 8. So majority of them are, or more than 60% of them are in the range of 4 to 6 ATP in each ring. So these are the 8 classes conformations we do. So because of that, we can now begin to map out which subunit in this particle has which one of these 8 conformations. So this movie shows how this is done. So first I want to show you how these 8 different conformations differ. It turned out the difference are at the apical regions. Of course, some have ATP and some don't in the enzymatic binding pockets. Now in here, I split that out again, each of these 8 subunit and assign for one particular particle where each of these conformations are. So that allows me to build a atomic model for this particular particle and do it again for the second particle and the third particle and the fourth particle. We have 165 particles, so that means we have 165 atomic models from this analysis. So I think the interesting aspect of this methodology is now cryo-EM is able to obtain not only an ensemble average like most of the structure you have seen, but now is the capability we can actually begin to look at the heterogeneous structures of almost identical but very similar but non-identical structure. As a result of that, we can build an atomic model for one particle at a time. And so with that, it concludes my second example. I'd like to move the third example. During the pandemic last year, we were all locked down. So I decided we would use the cryo-EM to look at different components of the coronavirus. So this is what the cartoon said in the newspaper. It's a sphere with some density sticking out, some spikes sticking out. So now there has been a lot of study that one can express the protein responsible to the apical region or the crown part of the spike, and then do either crystallography or single particle cryo-EM. So I think as latest statistics, more than 300 plus structures that was solved that way with cryo-EM of expressed protein of this spike protein. There are different types of protein that has been solved. So our research target is trying to look at the whole coronavirus and then see if we can restrict the structure of the spikes. And we also have projects that deal with the RNA piece of that as well, which Rachel Kretsch will present a talk tomorrow about the RNA molecule from the COVID particles. And so here is our data on the whole coronavirus. So this is a typical image of the coronavirus embedded in vitreous eyes. So what I circle out here are the apparent spikes that's sticking out from the surface of the coronavirus. The bottom panel are the the class averages of these box out particle spikes, and we box out 82,000 spikes particle from these, and we do the reconstructions. We were able to generate a 3.4 angstroms structure. This is the top view, and this is the side view. And then when we analyze the individual monomer, in fact, we have done both with C3 symmetry assume, and also with C1 symmetry. That means no symmetry in the reconstruction data, basically almost identical. So this is really a C4 symmetric trimer, even they are in the coronavirus. And so here is one of the protomer that extracted out that we're able to build a model. And in addition to the proteinous material, we see density sticking out, which interpret as the glycan. So before our work of extracting spike computers from the whole virus, David Wester in University of Washington in Seattle have solved a structure and expressed protein spike from this coronavirus. By the way, this coronavirus is an alpha coronavirus, which was the first coronavirus discovered in the early 2000s in Holland that caused only the mild respiratory diseases, because that was the only virus we could work on without the biosafety level 3 facility. And since we are working the whole virion. And so, so based on our structure, we actually compare almost very nicely with that solved by David Wessler with the expressed protein. And he was able to assign the density together with the mass spec data. So we match out very well with the assignment. In addition to those density that match with the expressed protein, we also see additional density elsewhere that are depicted in green color. It turned out that different virus growing in different cells would have different glycosylations consequence. In David's structures, his virus was grown in an insect cell, but in our case, it was grown in the mammalian cell. It's known in the mammalian cell the glycan structure is more branchy and complex, so that in that we did see it. Since the structure, we actually have done the mass spec. Now we have an assignment, basically all this glycan part with mass spec data. And so since we're able to see this density, I just want to show this called Q score, which is a numerical parameter which we assess the resolvability of the residue, both the amino acid as well as the glycan part. And so these are the amino acid residues. The Q score is way above expected Q score at this 3.4 angstrom, which were derived from over 50 PDB map that we analyzed. So using this Q score measurement of the density resolvability. Interestingly, when we apply this analysis for the glycan part, the glycan also we saw quite well with the similar score as the amino acids. And now, since we have the structure, since the most ultimate interest is how this alpha coronavirus NL63 differ from the COVID-2. So here is our structure on the top, and the residue number is different. The alpha coronavirus is slightly longer than the beta coronavirus. In the family, the virus classification, there's like an alpha beta is really different from this Greek alphabet nomenclature. Now they got delta coronavirus. Delta coronavirus basically is a derivative of this COVID-2, which correctly speaking is a beta types of coronavirus. But in any case, in both cases, they have the crown regions. They have two regions. One is called S1 region, another called S2 region. I call in the NL63 called S1, S2. In the COVID-2, we call S1 prime and S2 prime for the discussion purpose. Interestingly, that when we look at the COVID-2 spike protein, there in the S1 prime region, there's only 4 domains, A, B, C, and D. And in the alpha coronavirus NL63, they have 5 domains. When we compare the structures, when we superimpose each domain structures, that the A and A primes about a RMS deviation of 3.7. So the fold is really, really similar, but when we look at the sequence level, they're actually less than 10% similarity. But when we compare the domain 0 and A, that actually the tertiary fold appeared match very well. So that suggests that this 0 and A is probably gene replications. And so because of this structural similarity, so it's likely that they all come from the same common ancestral origins of coronavirus. So now the interesting domains of this, among all these domains, are the B domain, is where the receptor binding domain. Interestingly, in the NL63, A and B are in very tight interaction, as shown in here, all hydrotrophic interaction between the interface of the A domain and the B domain. Whereas in the case of the COVID-2, there is no direct interactions between the A prime and B prime domain. Okay, so that means in the COVID-2, the receptor binding protein domain is well exposed to outside. That's why the receptor can bind very easily and to trigger all the downstream consequence. Whereas in the alpha coronavirus, because the receptor binding domain is hidden in the regions where this interface between A domain and the B domain. Now, another interesting bit of the domain is the C and C prime domain. This is the only region, the only domain that has not only high structural similarity and also high sequence similarity as well. They're over 20% sequence homology. So I think if in terms of strategic thinking about how to build antiviral drug or the therapeutic drug, so that may be a good target because it kills not only the one family of the coronavirus, we may kill them all because this is highly conserved regions. In the other regions, in the S2 domain is the where the fusion domain, because after the binding that they have to fuse before the fusion take place. So they actually require another proteolysis. And so these two are not identical, particularly at the cleavage site, but they are very similar as well. Okay, so that's what we learned from this. So the take-home message I want to convey is now, one way in doing structure is to express and purify protein. As I demonstrate in this case, we can actually solve the structure in context without purifying or expressing the proteins. Okay, but using a computational procedures, we can actually extract a kind of near atomic resolution structures of spike proteins and of virus like the coronavirus. Now, so this is one approach to the problem. Another approach of this whole coronavirus is doing tomography, because I'm showing you a tomographic reconstructions of this NL63 particle. Now, as you can see in here, this is movie taken from a virtual reality graphic from a Chimera X illustrations, and you can see all the spike protein, as I showed in the previous slides, in deriving the 3.4 angstrom structures. The spikes are not distributed evenly on the surface of coronavirus. That means all the news we saw on television, all the spikes sticking out regularly. That's not true in real life. Okay, this is the case. Not only on the NL63, we actually have some data which collected in Galveston with about safety level 3 facility of the COVID-2, that actually the spikes also distribute irregularly on the surface of the COVID-2. So now in here, you can see all the coronavirus are actually sticking out somewhat different orientation with respect to the surface of the membrane of this virus. So we then use in other sets of computational method to extract the 3D volume of each of these spikes on the surface of the coronavirus. And so the next slide shows these we call subtomograph averages of the crown part of the spikes. So here is the top view, and this is the side view, and it's around 9 angstrom resolution by picking up a lot of this spike from the 3D volumes. And in addition to the crown part, which our cryo-EM structure only solve up to this top part, but not the stalk. The stalk part is right here. Based on the sequence, is predicted to be a coil-coil helix that form this triple helix to form the stalk. So since we pick up each of these regions from each of the spike, so having solved this every structure, we can put this back to each of the spikes. So as a result, some of the crown part is that perpendicular to the surface, and other one actually form an angle between the stalk and the crown. So that means they can be tilted. Some tilt less, some tilt more. So when we put a number of these pictures together, we interpolate all these snapshot, and we make a movie suggesting this crown part actually can bend. We see it looks like this hinge motion between the junction between the stalk and the bottom part of the crown. And then it's also have a twisting because they can have different orientation. So I think this approach provide, I think, a complementary information about the dynamic natures of these. So I think it open up all the possibility, particularly in the drug designs or antibody designs of the spike proteins of the COVID. I think this tomographic approach is another way of providing additional information. So these are the 3 examples I demonstrate how we can use a single particle. In the last example is a hybrid single particle and tomography of individual protein or RNA structures at near atomic resolution. Now, about 3 years ago, I was fortunate enough to be awarded a grant from NIGMS with the support from the Common Fund. So we are one of the 3 centers supported by this new initiative I set up called Stanford SLAC cryo-EM Center, which we have 2 missions. One is to provide state-of-the-art facility for the global community to collect data, which we run it like a synchrotron X-ray beamline. You apply with a demonstrated project and come to collect data either remotely or in person. And in addition, we sponsor a lot of projects to train individuals to learn cryo-EM at different parts, from specimen preparation, data collections, and image processing and modeling. And I think many crystallographers has actually come through our course and now become an independent investigator. And so last year, we set up our few dedicated 300 kilowatt microscope in our new building. And so one of the microscope have only a Falcon 4 detector. It's a brand new detector made by Thermo Fisher. The other 2, each of them has a K3 detector, more traditional detector. Well, I call traditional, it's only a few years old. And then with energy filter. So with this setup last summer, we decided we want to do a stress test exactly to see how far we can improve in terms of resolution. So we use our industry standards called apoferritin, similar to the ribosomes in the X-ray crystallography case. And so these are individual apoferritins, 480 kilodalton, and there's no iron inside, just only one protein that make up this protein cage with octahedral symmetry. This color represents the polypeptide, and this is they are made up of alpha helices. So this one alpha helix that with the side chain clearly, and also water and metal as well. So in this case, we actually collect the data only at 20 hours. We collect 588,000 particles, and we generate a map at this 1.27 angstroms. And so we use other microscope with other camera, we get around 1.34, 1.36. So this is around really close to atomic resolutions. And so, in here, we did a very thorough analysis, both visually and quantitatively, about the density. The panel on the left shows the 20 different amino acids, at each of them displayed in 2 contour level. And interestingly, you can see the aromatic side chain is actually a whole like donuts. And then the different contour level, you can make up individual atom density. Now, one thing I was told by senior crystallographer that in the old day, one would like to distinguish the density is the valine and threonine. In the valine, there's a 2 methyl group, they're more triangular shape. And then in the threonine, that actually have a hydroxyl group is a more round shape. So I think, again, cryo-EM density map does look like crystallographic map. They are really distinguishing different types of atoms. And then now in the charged atom, many of the structure has been shown in the literature or now in the PDB, so that the negatively charged residues do not show up the density, but at this 1.27, the density does shows up. That suggests that radiation damage is not the only possibility that one could not see the density at a low resolution cryo-EM map. So I think it has probably had to do with the negatively charged probability of this atom that responsible is absent in a lower resolution density map. And finally, we actually see there's a few of these residue that they look like to have 2 different conformers. So in addition, looking at it and enjoying the density, we can actually quantify, again, we call the Q score, which is the convolutions of each the atom density with respect to a reference density, which is a Gaussian with a small width. And so, and then we now routinely use this as a matrix to assess what the quality of the map at CRAM resolution at 1.27, and so compared to the crystal map, the Q score should be around the dotted line. So I think most of our density are above the dotted line. Of course, there are other regions that are higher mobility. Now, using this B score, the often asked question is, how does it relate to crystallographic B factor? So in order to put our B score in relationship to the more familiar parameter called B factor, we empirically derive this crystallographic B factor from our Q score in such a way that when we deposit our coordinate, we also deposit our Q score and the B factor in such a way that one pick up the coordinate, one calculate the map density does look like the experimental map. Like this one doesn't have the density, it does not have the density. If you don't take into account the Q score or the crystallographic B factor, and that shows up the density. And so I think this is one of the things we are promoting, that it is utterly important when we deposit our density map, not only the standard validation test, we need to describe our map in terms of Q score and also Q score derived B factor in such a way that the coordinate can faithfully reproduce a map that looks like an experimental map. And so here's another one that I don't have the time, because at that time when we solved the structure last year, there are 2 other labs in Europe, Hogerstad in Max Planck and Shaw at MRC, and they use a very expensive additional optics electron microscope, but their resolution is slightly better than ours, but not substantially. So in terms of Q-score and also the visual inspections. And finally, I want to make a comment about the detectability of the water in such a map. And so we do see that actually, that when we compare the water molecule between our 1.27 angstrom map with there's 70%, 75% of water are overlapping with the high resolution density map, 1.25 and 1.15. And so that is exciting because that means there are 3 independent labs determine the same macromolecular structures. They match not only at the protein level, but at the water level at 70%. The water position plus minus 0.5 angstrom uncertainty. And so I think that now testifies that the cryo-EM reached a stage that is a very reliable method down to the water molecule level. And finally, I'd like to remain a few minutes to mention some of the other important development in the field of cryo-EM. So I get into all the detail of all these near atomic and atomic resolution determinations. But what happens if we begin to look at things in the cell at a lower resolution between now, I would say between 4 to 0.4 nanometer. We can get down to 4 angstrom resolution with subtomogram averages. And of course, if the specimen is very thick, we can thin it with so-called focus ion beam to get a thin slice of a thick cell. So in other words, now the trend in the structural biology in the next decade is an integrated approach, seeing from not only the atom, but all the way of purified macromolecules to a macromolecule in the cell, and also with at the organelle level. I think that really signifies the excitement of cell structural biology that can put that in the hands of clinical science. And so now I just want to show you an example about this work we did some years ago of a cyanobacteria infected by cyanophages. This is a tomogram. You can view that in the microscope. And then in the reconstructions, we can, now this is a purified virus that can atomic resolutions. But here, now I'm now seeing these unfixed, frozen hydrated cyanobacteria infected with cyanophages at the early stage of the infections. And so you can see that there are lots of features, more than we are able to comprehend. So as a result, we need to really count very reliably through visualization as well as annotation. So now first, I show you some way of visualizing these. This turn out is a really threatened bacterial cell because the cell apparently have been lysed. A lot of content has been leaked out, so the contrast is actually quite good. But all the most of the content are still intact. These are the cell envelope. The cell wall is a gram-negative bacteria. It's a photosynthetic bacteria, so it's chloroplasts, and then also cyano. These are the Ribosome that form the clusters in here. And then the purple one are the newly synthesized bacterial phages. So these purple bits are all newly synthesized phages inside the bacteria. And the dark color one, we believe, are all different ribosomes and other molecular machines. And so here is interesting, we see these particular phages are going in this position. The tail are penetrating the cell wall where the additional density are facing outward. So I think that answers a long-standing question. What this horn used for? They're actually not for infections, but for different purposes. So we can actually, by looking at it, we're able to sort that out. As I pointed out, this is a cell that has been lysed. So now, based on this, we can look at the cell at early infections, intermediate stage infection, and late infection. Early infection, only phages outside. Intermediate, some phages are inside to replicate, and the late infection, and lots of new phages inside. So then again, we use these subtomogram averaging techniques allow us to average different types of subtomogram averages. We can actually find out 5 different particles that we can conclude they are made up of different stages of assembly. So you know, we don't need to do any genetic, just as Richard Feynman said, just look, and then we'll figure out some of these biological processes. And so finally, I kind of like to show one of the latest work we did with this Toxoplasma gondii, which is a parasite. It's a very rich. We're looking at the apical regions of these, and there's a different color represent different types of features. And then from that, we're able to do a annotation by some of the neural convolution neural net, as well as subtomogram averages. And I just want to highlight one of the recent analysis on this microtubule here. And we were able to do the subtomogram average, it's around 7 angstrom average. We were able to distinguish between the alpha and beta tubulin as compared to the structure done by Eva Logares and Ken Darling. And in addition, that we actually discover another protein inside the lumen of this microtubule that gas packet in a staircase. And so finally, there's another method that if the cell is too thick, we can use a thinning procedure, focus ion beam to make a thinner manner. And from there, we can actually see variety of subcellular organization. And finally, I'd like to conclude my talk by showing you a multiple example that the cryo-EM now is a high throughput procedures that can determine atomic resolution structure quite routinely if your macromolecule are purified and stable enough. And so our work is just like protein crystallography, a lot of web biochemistry has to be done in order to reach that atomic level. And I saw an example with the detector harmonized RNA that we can solve its structure without crystals. And we demonstrate with the ACA subprogram that we can build 1 atomic model per particle at a time. And then I quickly show that we can now look at molecular structure in situ without protein purification. And I believe that cryo-electron is an upcoming technique for cellular and physiological medicines. And finally, I'd like to acknowledge all my talented colleagues that were with me throughout the years. And then my collaborator, Riju Das on RNA, Freeman on subprogram, John Bufroy on the parasites, and John King on the bacteriophages, and Steve Lukic, who used to work with me, continue to give hands on the data processing. I'd like to thank you very much for your attention.
Thanks so much. Wow, that was amazing. I wish we could spend another 2 hours just on the cryo-ET part. So when you were looking at the virus, that was amazing that you were able to do that, by the way. And you saw the dynamics of the spike protein that you showed us. Do you guys see any movements within the membrane? So in other words, sort of clustering of spike proteins?
Yeah, that's a good question. We haven't gone to that. That's a good question. That's the next course of action.
Yeah,
of course.
yeah, because I know there is a fair amount.
Yeah,
there's some fluidity, right. I know. So
yeah, right. So yeah, I think Jen has her hands up. Professor.
Oh, sorry, Jane. Yes, go ahead. I didn't see your hand. Please.
All right. I was really excited by your ability to visualize the individual particles, and I was wondering, one of the cases that we really don't understand very well is, for instance, the polymerase with the remdesivir bound. The remdesivir is only there about half the time. And it would be wonderful if you could go back to the individual particles and separate out the ones with and without, and see whether you could really visualize these 2 states of the active site, because the overall conformation doesn't change much.
Right.
Maybe you could separate them.
Absolutely. I think this is really open up a lot of possibility. We first demonstrated with the GrowEL some years ago. So just a pure GrowEL, and we were able to pick up 3 conformations. And interestingly enough, these 3 conformations has corresponding crystal structure where crystals are different at different time by in different lab. So that means what we are seeing is really energetically feasible. And so right now, I think with your suggestions that extended to polymerase and other molecule to look at individual particle is right on the dot. Yeah, so there's one question from Dr. Ma about the chaperone, about the ATP occupancies.
Right.
So I think the interesting thing, what I am alerting to in that example is really a kind of close to continuous conformational variation. So even though I classify 8 classes, because that's what we asked for, and they give me 8 classes. Now, if we expand the data to a higher data set, okay, and we find a classification, I think we can possibly to see more conformations. I think one of the things we are now doing is actually to use the molecular dynamics to start with one conformations, and then let it run for days and months, and then see where it land up, because we have multiple states now as an individual subunit. So I think we can actually track that. And I think this is, I think the way to sort this out, I believe it will be much easier if we involve the next generation of image processing methods. Okay, I think what we have done is very great from what Reliant and CryoSpark provide us is already can get us there. But in order to get a fine-grained separation of these dynamics, which the cryo-EM investigator like myself begin to understand the work dynamics. So I think, and also, and the image processing developer understand what the biology. I think this will be then other future directions to get after that. That need a integration with 2 fields.